Thank you, Julia. Thank you very much for the invitation. I would also like to thank you for organizing a lot of interesting meetings with very many people in the last 2 days, actually. So I met with Harry Landesmaki from Aalto, Willem Student from Helsinki, Alexi Callo from CSC, Samuri Patti, and Mitya Kurki and Andrea Ganna from FIM. So I had to read these names because unfortunately with age it's very hard to read the names. But these are very important contacts which also made me understand that there is a live community in Finland, small country, and a lot of genomic work going on. So maybe the country with the highest percentage of genomic work per person at the moment, as far as I can tell. So when I give lectures on this topic, typically, the public divides into two categories, the one that never heard about genomic computing and the one that know everything about genomic computing much more than me, because I started, like Giulio said, only 5 years ago. And normally, I make the first community very happy for the first half, because they come to know a totally new field, and then they sort of end up being lost in the end because they start to give some technical detail. And the second community is totally unhappy in the first part because they know everything, and maybe they get something out of the talk at the end. This time I'm trying something different. I'm trying to move in the middle something which is very generic as well, such as cancer research. So at a very broad, in a very broad perspective. So maybe I will interest the people who know nothing about genomics up to some point, and then interest the people who know nothing about cancer research from some point on, and make somehow the community is happier than usual. That's my hope. That's how I plan the talk. So let me start. Today we are talking about genomics. This is a map that everybody shows when we start talking about genomics. It's the cost of sequencing. You may see that the cost of sequencing has gone down tremendously from, let's say, the cost of sequencing the first genome, which was around year 2000, up to the current level, which is less than $1,000. Yesterday at FIM, I have seen one of the machines that can do genome sequencing for less than $1,000 per genome. So the turning point is, of course, somewhere around 2007, 2008, when a new technology came out, which is called next generation sequencing, where the cost of sequencing has started to drop much faster than the Moore law. On the other hand, when I consider the genome, and after these few years, I mean, it has more or less the same amount of information as a Britannica encyclopedia, and I put the version in Chinese because I don't understand Chinese, much in the same way as I don't understand the genome. So it is very complicated to read all this amount of information. And so finally, to introduce the topic, I put here Alan Turing's face. You have seen for sure the movie, The Imitation Game. And essentially, you know that Alan Turing, among many, many other things, he also helped to win the World War by deciphering Enigma. So I think we, as computer science, are facing, say, a task which is as difficult as, or probably much more difficult than the task that was originally, let's say, faced by Alan Turing. The fact is that he had very, very, very different computing power. So now we have much more computing power, and we are facing a much more difficult task. So genomic, let's say the start point for the kind of work that I am interested in is the sequencing of the human genome, which, as I said, happened in the early 2000 years. And you can see that it was a result of a lot of disciplines, including engineering, chemistry, biology, physics, ethics, and informatics. So it is really a multidisciplinary task. We can see how much it is growing in terms of the, let's say, data, the expectation about how much data will be produced in a short horizon of time. This publication of 2015 indicates that we expect to have more genomic data than any other science. Currently, astronomy is the strongest data producer. We are currently expecting to have more genomic data than will be on YouTube. So thinking about whoever video is going to be uploaded to the media. And also you can see that there is a lot of, let's say, computing intensive activity which has to do with genomics. So it is not just a matter of putting data there, it's also a matter of being able to let's say, analyze it, calling the various features. By the way, the title which I gave, Deciphering the Signal from the Genome, has to do with the fact that we look at the genome no longer as, let's say, a sequence of symbols from a computer science perspective, but as a sequence of signals that these simple symbols produce after being a lot of analysis that I'm going to talk about. So integration of signals really means integration of, let's say, aspects which we derive from the genome and describe it in abstract terms. That's my point of view. So it's becoming closer to the problem that Alan Turing was facing. This is a description of how genomic, let's say, data analysis is structured. And again, it's not a slide which I produce. It's taken from the web. Essentially, it says that whatever occurs in terms of data processing in genomics really can be separated into three phases, where the first one has to do with essentially producing the sequences. The second one has to do with a variety of techniques which I assimilate to calling. Calling in genomics means really producing. We can call mutation, we can call peaks, we can call, so extracting signals. And then the third phase is what I'm mostly concerned with. It is, and to me, this third phase really looks like data integration. I've been looking in data integration in many domains, and in particular, the data integration problems which are applied to genomics deal with annotation and filtering of variants, data aggregation, association analysis. These terms, to me, assimilate to the broader, let's say, discipline of data integration. Now, the situation is as follows. While primary analysis is essentially done at the machine which generates the data, secondary analysis is essentially very rich in techniques which have been produced by the bio-informatics community in the last 15 years. There is still not much going on in tertiary data analysis. Here I list the system which I consider to be competitor to the system which I'm developing, but there are not that many. Maybe it's because the field is still immature, because at the moment there are not that much big data that can be considered by looking at the signals which are made available by the international community of scientists. But this is going to change because projects such as the one which is taking place at FIM, where 10% of the genomes of Finnish population will be sequenced, or the projects which are taking place in UK when we are talking about 100,000 genomes. And anyway, many other big population sequencing projects which are ongoing are essentially going, in my opinion, to bring more interest and more importance to this area of tertiary data analysis, where, as I said, the players are relatively few. So another thing which, again, is to please only the people that are very new to genomics. Which kind of signal are we talking about? We are talking about variants. A variant in a single position can cause a lot of damage. This is in particular the case of genetic disease. This is a mutation which create a very known sickle cell anemia, which is a very known genetic disease inherited. But there are also other signals. For instance, if you see this diagram. This diagram shows 4 genes, which several genes in 4 different conditions, and these genes can be either active or inactive. The green color means active, and the red color means inactive. So depending on the specific condition or tissue, or we can talk about whether the gene transcribes or not. And also we typically look at signals which are collected along the DNA, because there is some kind of reaction which takes place, which is such that the reads accumulate in specific positions, and then they form sort of peaks of reads. And then these are additional signals that indicate how the DNA reacts to specific treatments. So all in all, these techniques correspond to, let's say, the most important. This signal corresponds to the most important techniques which are let's say, performed by next generation sequencing machines, which have to do with looking for variations, looking for gene expression, gene activity, looking for the protein-DNA interactions. And correspondingly, they are called DNA-seq, RNA-seq, and ChIP-seq. So how would such signal be presented on this instrument, which is a genome browser, which is what a typical biologist would use in order to read these signals? Well, essentially, a biologist would look for things such as, let's say, the position of the genes. Let's say here, this black are position of the genes in the genome. And then it will look for whether these genes are expressed or not. So this red signal indicate the gene expression. Then would also look for experimental data set which can be interpreted as peaks of expression. And then we look for a specific position where we have mutation. So you could ask things such as, give me the expressed genes where there are enough experiments which confirm the existence of some biological effect, and where you have also find mutation. This would mean finding interesting area on the genome. I'm going to come back to this example in a minute and show how our language makes it possible to locate a region of this kind. But let me do, let's say here, let's say a definition of what is the project that Giulio mentioned. So I am working on this ERC grant since about 2.5 years. Actually, it's just I'm just started the second half on the 1st of May. The focus is on data management, integration, and structure analysis of tertiary genomic data. What we would like to do is essentially to try to bring a change in the abstractions, make them at the same time broader and simpler. So what we would like to do is to design very, very simple languages by means of which it is possible to do data extraction and data and prepare for data analysis. I hope I will convince you that by the end of the talk that this is we are along the right track for doing this. Of course, everything has to be open source. And what we have done so far is to deliver the first prototypes, let's say, that essentially go along those claims. So in a nutshell, this is what we do. We assume that there is somewhere a biologist or a bioinformatician or a computational biologist who have some needs. Well, first of all, it might find useful to look at open data. And so there are a number of places where open data can be found, because there are a number of consortia that collect open data, and they are collecting data for secondary research use, which means anybody can do whatever he wants with the data which are available to the community. And in particular, I cite some names. ENCODE is the Encyclopedia of Genomic Elements, TCGA, the Cancer Genome Atlas. So you understand from these names that essentially it is these collections are trying to make a wide mapping of the information which are available for doing genomic studies. So a first result of our work is to a system that essentially collects information from all this data. I'm going to talk about it later on in my presentation. Through a systematic chain of transformation, the metadata which describe these experiments get normalized and enriched in a way that it is possible to query them as if they were creating a single data collection. And at the same time, we also load the process data into a repository. This repository sits in Cineca, which is the supercomputing system in Italy. It also sits in our servers in Milano. So essentially, whoever wants to use our system has access to the process data which are produced by, at the moment, 8 data sources. And we are going on slowly. We have a machinery that we can use to integrate more data slowly and slowly. Then this is the main thing that we developed. This is, let's say, a language which allows you to go on this data, process them, and extract the important and useful data. Why? Well, essentially, the job doesn't finish here, because when you have an interesting region, you still don't know what to do with these regions. But you can load them into data analysis tools. And you can start a second part of your work, which has to do with analyzing the data. The fact that this system is, let's say, compact and simple to use and so on, will make the cycle of going back to the data, doing the analysis, much simpler than it would be. Let's say, we will try to simplify this part. So the user can essentially use these two interfaces, one for locating the data and the other one for actually using the data. Typically, the data which are located are also available to the computing environment. And also, the idea is that we are working on how to federate several of these boxes. And you see Spark here is an element of this box, because what we do, we essentially read and use the data from a variety of interfaces, including interfaces for R, Python, and Galaxy, and Firecloud. We have also a web interface. Whatever we do eventually end up being executed on Spark. So essentially, the abstractions, because I told you I want to build abstractions, the 3 abstractions are essentially a general data model by means of which we can describe any source of data, chipsec, RNA-seq, or additional types that we are also considering. A language for expressing operation on them. And then a conceptual model for the metadata that we use in order to instruct our repository about term matching and term transformations. So, and to the main message which I have given with my ERC proposal was, let's try to see if by means of this abstraction it is possible to introduce a simpler and broader processing technology. Okay, so now I will go for about 10 minutes describing to you the method, and then I will go up again and try to describe application that we can do by using the tools. The model is essentially a model where we have regions and metadata. For regions, the regions have essentially coordinates. So they sit in a given chromosome, they have a given stop and start, they have a given strand, and that's it. So every signal that we consider has this component. And then we have a feature vector that can be anything. At this point, this is the simplest possible feature vector, which is just the p-value of a particular peak, but it can be arbitrarily complex. And then we have metadata pairs, which describe the metadata that each file or sample or is taking with together. So the idea that, for instance, this is a collection of, I don't know, expression data, for instance, for a particular patient which has a given tumor and a given age. And these are the only data that I have in this specific situation, but I could have hundreds of attribute value pairs. These things, files of information with exactly the same structure, are what we call dataset. So, for instance, encode narrow peaks are for us a dataset, which has several thousand samples. And we process these things in our language so that the result of operation also includes metadata. So that by doing operation one after the other, at the end of the result, you can read the metadata which come from the original sources that we have considered. This is a bit more for the bio-informaticians, but they will essentially find the format, typical format for describing mutation or expression data or ChIP-seq as a feature vector that we combine with the regions. Okay, then the query language. I give you an intuition of a query language by essentially doing the query that I have mentioned to you on the browser by means of our language. So what the language would do? Well, first of all, it would say, well, first of all, what the model would do. The model would reduce these signals to regions. So the regions are essentially a start, a stop, an end, and some features. So the peaks are reduced to their height, their width, their strength, the p-value, whatever. So in the end, we reduce each of these signals to features which are localized into regions. For mutation, it's simple. I mean, the region is a single base. But of course, we have also other kinds of data or kinds of mutations, that's insertion or deletion, that require specific, say, care in being positioned on the genome. But that's not a big problem. Then the query about giving us the peaks which are confirmed over at least 2 experiments. You see that there are 3 experiments here, but some experiment could be faulty because essentially the data could be missing. So what we are asking to our system is, okay, we have 3 experiments. Let's say that the peak is there if it is confirmed by at least two peaks. So our instruction for that is cover. Cover to any, so the 2 to an arbitrary number of the ChIP-seq data will give us the confirmed peaks. And then our query for asking which one are, let's say, together with, are intersecting a region which is expressed. Well, we take an NSEC data, we take the peak data, we join them, distance less than 0 means intersection, and we take the left, which is the peaks. So we project on the peaks whatever is produced by this join operation. And you see that, by the way, we take away 2 sets of region by doing this join because they don't match. And finally, we say, okay, we also join with the mutation. Same thing, we take the result of this, which is called S2. We take the mutation file, we join by distance less than 0. This time we take the output on the right, which means we preserve the mutation. So the result is this one. Okay? So in general, we have a language which accepts a lot of inputs from different, let's say, signal of different, let's say, types, we say, or different signature, or different, coming from different experimental techniques. We support several operations, out of which many of them are conventional, let's say, data operations, such as grouping, selection, projection. Some of them are more specific to our genomic work. So we define genometric join, map, and cover. And they work on big data. So what the data means in this case is relatively small because I'm talking about thousands of samples and maybe millions of regions. But nevertheless, the performances are, and this is actually kind of old. The performances are now better. But the performances are order of minutes in doing this overall encode, this kind of processing which have to do with joins or mapping or cover that are the typical operation that requires combining pairs of let's say, of samples. So they are the heavy ones. A final operation typically produces, and which is called map. I don't want to discuss this because I don't want to go in great details about the language. But the typical operation, what it does is to produce some kind of a map where we have experiment on one side, regions on another side, and a feature vector in between. That can be interpreted as a matrix. And at this point, whatever you can do with a matrix is done by means of classical libraries. So we can produce a need map, can interpret it as a connection network, or, which is most typical, you can take a library and then go and do additional machine learning or deep learning studies by combining with whatever library you can think of. We take these as expression data of particular regions which are genes, and then you find additional interesting properties about your genes. So this is outside our scope. Our scope is extracting the nice collection out of the existing data and then letting anyone applying whatever data analysis method he would like. How to do this? Well, essentially we have a web-based interface where you can go ask. This is exactly the same query, similar query to the one that we expressed. You see that on one side you put the query, on the other side you have access either to private or to public data. Incidentally, you can connect to the system as a demo user, so you can go and try the system without requiring any login, any, let's say, additional thing. If you want to go and try, you get not too much resources, because of course we don't give too much resources to anybody who just come and log in, but enough to try simple queries. And you can browse the data, you can see the metadata, you can see the schema, you can have a lot of facilities to help you to understand what is going on. However, my students typically don't use the web interface, they use the Python interface. So what the Python interface does is essentially to reproduce the commands one by one with exactly the same logic as in relational terms. At some point you say materialize, and this starts and produces the result. And the result is then put into particular data frames where it can be used by using any other library. So it can be used using pandas, it can be used using deep learning libraries, machine learning libraries. So then the beauty of this, and I'm going to come back to this at the end of my talk, is that if you have everything included in a notebook, the interplay between changing parameters, doing the experiment, viewing the result, and doing the analysis and back becomes a bit easier. I mean, it is, I think, what the data scientists are becoming accustomed to do. A notebook where they essentially store their data science work. Another way to look at our system is essentially by means of what we call web services, in which case, essentially, you don't see the system. This is a web service that has been developed for the integration of transcription factors. And what we wanted to see is whether transcription factors interact, they cooperate. So the idea is that by means of this tool is trained over the entire encode to understand something such as the typical distribution of distances of interaction between transcription factors. Then you can give your own data and you can see whether it is according to the same metrics. You can prove or disprove, let's say, the interaction between transcription factors. So I don't want to say much, except the fact that here you don't even need to know that there is GMQL below. GMQL works and produces a result. This is just a web service. And another thing that we did, we installed GMQL at the Broad Institute as part of FireCloud. So FireCloud is a workflow flow system where you can essentially do very heavy computation that is offered inside the Broad in order to provide Google computing facilities and Google and also, let's say, Google storage, and also as an accounting mechanism that everyone who is launching a big data computation knows what is going to pay, because it's going to pay something. To whom? To Google. And the idea is that to the Google Cloud. And the idea is that here we have what is called a featured workspace of the next generation system which is coming out, which is called Terra. It is a next generation of FireCloud, where by trial and error you can essentially replicate work by using our own system. And we are trying to improve the way in which this work could be done. We are progressively in touch with the people who are developing FireCloud and developing notebooks on top of FireCloud, so that this collaboration may bring to a better use of our system inside the FireCloud. I already talked about the repository. The repository uses a conceptual model. Well, I will give you the idea of the model. The model has a total of about 30 attributes, so it is very small. It is centered on the notion of item or file or sample. And around it, we say how it is biologically produced. So we have what is called the biological view. Then how it has been produced from a technology, so the technology view. How it is put into data sets, so the computing science point of view. And how it is managed, which project has developed this result, under which circumstances, it's a code project, or how the projects are grouped into cases or things like that. So this was a terrible effort, because we started from ENCODE, TCGA, epigenetic roadmap, ICDC, and they have different description, but it is possible to map their description to this by data transformation. So this is what we do. We do loading and transforming, then cleaning, then mapping, then normalization, and then also have some integrity constraint checking. So that by the end of this chain, whatever we put is relatively, quote unquote, harmonized between the various sources. These are the numbers that we have at the moment. We have 240,000 items coming from 6 sources. And this is not yet available, but if you're interested, I can give the link to this, which is becoming available. It's relatively completed. And it is a system that allows you essentially to put out the various attributes. Here you see that the attribute is instantiated with a possible value. Prostate adenocarcinoma, PRAD, and the assembly. And then if we run this, we will see a given number of items found, which are spread over several data collection, which are the data collection we talk about PRAD. And you can go item by item. You find a link to the source and the link to our internal representation. If by the end of a story you have, maybe it's a little bit big, but you want to do a query which involves these 4,800 files, well, you can do it. They are homogeneous in type. The query will be generated automatically, because the query will be a big select from these 4,800 things, which will be shipped to our engine, and you can do some of course, maybe in this case it doesn't make sense because we are mixing ChIP-seq and DNA-seq, but maybe not. Who knows? I mean, anyway, this is in theory possible because it is all compatible, so to speak. As I said, the data architecture is based on Spark. It is a relatively complex system with a lot of layers. I will be relatively short because I see the time is going by very fast. We have an intermediate level this, which is called directed cyclic lock, is not very clever as a name. It's just the name of a graph. But this graph describes the operation in a way that is independent from the source. So it can be run from GMQL, can be also, it is an API that is used by Python or by Galaxy or by R interfaces. And below, we had, over time, many alternative sources. And at the moment, we concentrate on Spark. But we have a Spark to Flink comparison, a Spark to SciDB comparison, which were published in conferences. If you look at what happens, well, essentially we map GMQL to the DAG, then we can do some kind of optimization, which are somehow interesting. And there are interesting papers. If you're interested, I can point you. All the papers are available in our website. Then so in this way, we optimize the DAG. Then we go to the implementation. The implementation technology, I will say a few words on it, is based on the notion of binning, because we have to bin the genome into slices, and then to be able to perform parallel computing over the slices. And the idea is that you bin the genome into portions. You do in each portion computations such as join, map, cover, the one that we defined. And then in the end, you aggregate the result. And the typical profile of what happens with execution engines, where we are talking about clouds which can scale, or operations which are different, and so on, is that if you take a specific operation, okay, normally there is a sweet point of execution, which is the best possible bin, because if you move away and have larger bin, the operation inside bin are too complex. If you move in this direction, you have too many bins. So essentially, we have done the optimization and all the mathematics which corresponds to implementing in an optimal way our operations. Okay, so up to this point, more or less where I wanted to be. I have only 2 minutes delay, so it's not too bad. Let's talk about what is the most interesting application for genomics. And of course, it is cancer. So now here I am not at all a cancer expert, but I was at the luxury of having a cancer expert invited to a workshop, which I did a week ago. So this is taken from experts of the domain. It gives you some numbers which are important, such as the new cases in Europe and new cases per day in Italy, the diagnosis and the prognosis, and how many can be cured. Cure means typically living after cancer in a given period of time. This is a description that says essentially the probability of survival after 5 years. It shows that it has changed a lot. It is for the United States, and it has gone, essentially the difference is about 40 years, 37 years, going from 50% to 67% overall cancer. And of course, you see that there are, let's say, cancer types which are more, let's say, benignos, in the sense that they bring more easily to higher survival rate. And there are some other cancer types which are extremely bad to care and to resolve. This is the diagram for the US. The diagram for Italy is not very different. There are differences in numbers which are 2-3%, but essentially they are not, they're essentially the same distribution and the same also degree of progress. This marks a little bit the distinction between, and I think healthcare in Italy is not too bad. So it's a little bit the distinction between a typical healthcare system in Europe and a typical healthcare in cancer, which is supposedly very well developed in the US. And here is where, let's say, cancer is distributed in terms of incidence, male and female, and in terms of mortality. By the way, I'm giving you a good message here. Finland doesn't have a bad mortality. It's overall being both for males and for females. Females have slightly higher incidence, but in the end have the same amount in terms of mortality. This is another message that you can take home, because I think it's not too bad compared to the rest of Europe and the rest of the world. So why am I talking about cancer? Well, I'm talking about cancer because there is something which is called precision medicine, which is essentially the idea that you give to each cancer patient a specific treatment. And this is not only related to treatment, it's also related to the fact that if you have a diagnostic mechanism, you can do prevention. And in particularly, primary prevention has to do with avoiding the disease, while this has mostly to do with human habits. Smoking, being exposed to sun, whatever, or to pollution. But secondary prevention is whatever you do when you have a first sign of cancer and brings to early diagnosis. And yet there are techniques that you can specifically do when you see specific signals in the genome that tell you have to be more careful. Or a surveillance program, which means you had cancer and now you have treated it, but we want to make sure that you will not have relapse. So we want, or we want to anticipate relapse to treat before. So there are programs in which I actually made some proposals that are not yet being evaluated, where having some genomic information can help not only the therapy but also the prevention. Just to give you an example of a problem that fascinated me and where I would like to see my people involved more and more. This is a study about ovarian cancer, which is taking place at Mario Negri Institute, where essentially the idea is that ovarian cancer is a very bad disease, but it is specifically bad for the population which is called as a genetic alteration which give resistance to treatment. Because if the population is resistant to treatment, it means that you give them the chemotherapy and nothing happens. Essentially, the person dies because there is no reduction of the cancer cell proliferation. So the kind of research in which we are involved, this has actually been done by biologists of Mario Negri. We are only doing a refinement of the methods which have to do with detection. It's an early detection of in which direction we are going. If you do the detection here and you belong to this part, instead of doing the chemotherapy, you can do as an alternative therapy. Okay. So this is something where we see the possibility of really doing something significant at the level of really at the what is called translational approach. They give it right away to the patient. And this, we have a population of resistant patients and sensitive patients, and we are essentially trying to build a classifier that can predict. And the reality, what we have some candidate genes that generate the classifier, and we also a nice interplace between the public data set that we take from TCGA and private data set that we take from Mario Negri, in order to build a good use of the data. I mean, use the data for validation purposes, the one that we have produced at home, and make sure that this separation actually is validated. There are more complex studies. One study which we are doing has to do with it's a little bit complicated to explain unless you have the notion already, but essentially the genome binds itself and creates three-dimensional loops. So we are studying what these loops are actually, the loop is somehow represented in the abstract as something here that goes from a given coordinate to another given coordinate, and actually this one. And the idea is that at the end of the loop there are typically certain proteins, and the protein that we are looking is CTCF, which is the most important protein to create the premises for this loop to be effective. And we also take the ChIP-seq signal that we have been discussing before to see whether this protein in the specific tissue where we have also the loop is, let's say, is active or not, it has a peak or not. And so we look at the boundaries of the loops, and we look at the position of the CTCF protein, and we look at the fact that they are active or not, in order to build a subsystem, which then we analyze from the point of view of various cancer types, because in this specific position of the CTCF binding, there could be a mutation. And the effect of a mutation is essentially to open up the binding in a way that the functional effect of these loops, that is to isolate the members of the elements which are in the loop, is no longer valid. And though you could have a situation where there is proliferation, for instance, because enhancers come in touch with genes while they are not supposed to do so. And this was published in a paper in Nature in 2016. And there are a few other examples of this kind. So we are trying to do a systematic study. We took 27 tumor types, 3 cell lines, looked into data from mutation, methylation, and copy number alteration, which means essentially we look at the super big system, all from open data, or from public data. And for instance, this is an example that shows that the mutations are enriched when we look at the active boundaries. So essentially, these boundaries are attracting mutations. And this may mean that the mutation, which is, let's say, normally, let's say, a difficult process to understand, in the specific case of cancer alteration, will concentrate on that specific point, which is a point where there is some destruction. So we did, our results are a classification of motifs, analysis of mutation, permutation test to assess whether these mutations are outside let's say, the typical values. And we use our language. Here, this is an excerpt of our language, where essentially we can say, okay, we build a collection for doing the permutation tests, for doing the visualization. We put there a tumor type, we put there a particular feature, we put there certain parameters, and we create another situation where we can do this testing. So this gives you an idea, an intuition of where the language could bring you. If this is embedded into a Python notebook and it is somehow well understood by the biologist, or has been, let's say, worked out with some kind of good, clean design, then it can be massively applied to the various situations. And with about 20 lines of GeMQL query, we systematically looked at 3 sets of neighborhoods, 26 tumor types, 3 genomic features. And the results are somehow interesting and currently under review. Let me finish the presentation with 2 considerations, which again pop up a little bit. So first of all, this just appeared on eLife. It's a recent paper, and show the fact that the data science approach could essentially be used in the various phases of experimental work. You can use it for generating the hypothesis. You can do for design the experiment, performing the experiment, and then evaluate the result. Data science is important in every possible parts for generating hypotheses, for doing fast feedback and optimizing parameters, and eventually making the results shared and accessible and interoperable. So it is an important message that given that we are talking about the data science community. The data science community in experimental work should keep in mind this principle. And the last thing I want to say is, then we'll come to the conclusion, is that there is an important aspect that has to do with reproducibility of result, that you would like not only to find the result, but also to be able to reproduce them when you are... reproducibility is not just this, it has also to do with the fact that there was a very nice talk last week. Essentially, you have to also keep track of which version of which system and which component, which library, and so on we have been using in order to do the testing, because it is very delicate to understand, to set up, to reproduce, that is to say, to redo the same experiment maybe 3 years after, if the setting has changed in between. Reproducibility is helped by using this kind of instrument. That is to say, a single environment where if you essentially use libraries which are well known in terms of their identity, and you track very well what you are doing, well, then you can essentially take the library itself, let's say the notebook itself, and use it as a documentation of your work. And maybe submit the notebook together with the data. So there are interesting things here that have to do with, as I said, the data science and the reproducibility of experimental work, in which we are not, of course, at all the expert. We are just pointing at this as important problems. So as a conclusion, we have the GMQL system, we have a repository, we have done several applications. This is my team. I have essentially a co-PI. I have 8 PhD students. I have 2 postdocs, several postdocs which have been scattered around the world. This is a relatively recent team building, because as I said, I moved into this field only 6 years ago. These are the master graduates that we had. These are the collaboration that we had or we are having. And so it's a group which is trying to give contribution in this field. We have published the query language, the data model, the conceptual model, and the big data management systems. Typically, the most interesting publication appear on bio-informatics. But for instance, the publication about genome binning or indexing and stuff are in typical computer science journal, or metadata management and so on. And you can find more stuff in our website, where essentially there is a provocative page that says try GMQL, where you can find also these are the manuals, introduction to the language, example query, biological examples. You can find the REST API, the GitHub site, the web interface. You can download, and you can go to the Python or R or FireCloud environments. Okay, so I hope you will have some interest in doing this. And I'm here for getting a few minutes for questions. Thank you for your attention.
Thank you, Stefano. Really, really nice talk. Now we have a few minutes for some questions. We can then continue the questions also outside with some refreshments. But let's take, who would like to break the ice? Who has enough courage to break the ice? Let us show that the Finnish audience is indeed...
good. Then I will tell you a story about breaking the ice in Finland.
Yes. Finnish version, of course. Just kind of out of interest, do you aware of any other domain-specific languages which were like designed for use in bioinformatics?
What do you mean by domain-specific language?
Well, like your core language is specifically designed to work with genomes. Do you know any other like languages which are specifically designed to work with genomes?
Yes. But then the question is what?
If you know other.
If I know other languages like this. Okay, yes. There are a few. So first of all, there is something which is called GQL, so that we have to say put an M in between, as a genomic query language, which has been developed in San Diego, and it's been out for some years. There are, I think the Blueprint Consortium has some rudimental query language for doing some of the operations. Of course, if you go at the level of, let's say, libraries for doing, for embedding your work into program, into, let's say, scripts, the kind of operation that we do for comparing regions, and you have BedOps, BedTools that are specifically bioinformaticians oriented libraries, but they operate on a pair by pair of the sample. So you have to build around it all the control which has to do with for essentially bringing in iteration. So you have to put, if I want to simplify it to maximum, say that you can build some predicate that would extract the exact regions from a pair, then you have to build let's say the workflow that would iterate over all the pairs of one and then over all the pairs of the other. But essentially then you can programmatically write very similar things. However, you don't get the same performance in terms of cloud-based computing, because we can combine on a cloud very many of these operations, while this is maybe parallelizable, but has not been thought in order to run on a cloud. It runs, I think, it's implemented in C, and it runs on a single, on your machine, so to speak. Yeah?
I just wondered whether your software has been used for splicing analysis, alternative splicing analysis?
No.
Oh, okay.
No,
do you think it is possible to use it?
Okay, so why not? The thing is that it has to be expressed in the terms of queries that would give us inputs and output in a format that we would accept. So you are welcome to look at this problem from this perspective and see if it can. Of course, it also depends on which kind of query you have. So what kind of regions you want to find when you have alternative splicing. So
is it possible to filter the reads to include only the intron spanning reads or exon junctions?
I think that you need to give essentially information about intron-exon structure. And then you have to think about what is interesting in terms of intron-exon relationship and what you want to find and how you would like to express this in alternative splicing. And then I think you can come up with interesting queries.
Thank you.
But the data type for doing this, we don't have. We didn't consider so far, but can be, of course, region-based. Okay.
More question, or is it time for your anecdote on breaking the ice?
Well, the anecdote was that a long time ago, in '86, I was giving a course in distributed database system, and this was in Finland, actually in a place called Karuela, which was in the middle of Finland. And I was giving this course, and I think the course was going pretty fine, but I couldn't get the attention of my class. It was terrible. So I was saying, what is wrong here? And then at some point they said, at the end of the course, they said, Professor, we all go to the sauna. Do you want to come with us? So we were all there. I mean, really all. And when we came back, there were beers, and they started to make questions. And I had a huge amount of questions. I couldn't have made more questions than in this specific event. And from this point, I have 3 houses in Italy, and they all have a sauna. So it means that I have become an addict to this event, and Giulio knows.
Yes.
Okay.
Thank you, Stefano. I just would like to add that the Finnish audience is very attentive. It's just that probably we have the cocktail now, so a bit of sparkling wine from Italy. And next time we will serve it before the lecture. So then.
Okay. All right. So thank you very much again.
Thank you. Thank you again.